one of the type of immunity. We have uh, two types of immunity, that is innate immunity, as we have discussed earlier. Immunity is divided into two types, especially. One is that innate immunity, another one is that adaptive immunity. Innate immunity is that it is being developed from by birth. By birth, it has been developed by birth. And coming to the adaptive immunity, whenever our body encountered against different types of pathogens or parasites, then our immune cells, interaction between our immune cells and immune organs, immune molecules with foreign pathogens and parasites will develop the adaptive immunity. After exposure to the uh, foreign invaders, then only adaptive immunity will be developed. Then in this adaptive immunity, we have two types of subtypes. That is, one type is that humoral immunity. We have talked about this humoral immunity as a unit 6. And we have another subtype of immunity under the acquired immunity, that is cell mediated. Humoral immunity is a branch of acquired or adaptive immunity, takes place with the help of antibody only. Okay, wherever the antibody actively involved in the neutralization of the foreign invaders or antigens, that type of immunity is can be called as humoral. In this type of immunity, only antibodies are actively involved. Uh, Coming to the cell mediated immunity, in this type of cell mediated immunity branch, in this immunity, cells, specialized cells are there for dealing with this type of cell mediated immunity, especially lymphoid cells. Other than lymphoid cells are actively involved in the innate immunity. Just remember this point. Other than lymphoid cells, I am talking about especially lymphoid cells. Okay, lymphoid cells only involved in this type of cell mediated immunity. Other than lymphoid cells are involved in the innate immunity. Directly or indirectly, these innate and adaptive immune cells are interlinked and combinedly function to encounter the foreign pathogens or parasites. But coming to the innate immunity, other than lymphoid cells are involved. Only lymphoid cells, that is T cell, T lymphoid cell, and B lymphocytes are actively involved in this type of cell-mediated immunity, which is comes under adaptive immunity. These all aspects are we have discussed in the offline classes. Okay, those who have not uh, come to the this class, those who are not there in the class, uh, I am just briefing the previous classes. And after that, we have discussed the complement system is one of the innate branch immunity. The complements proteins, the specialized proteins are there in our immune system. The proteins which are actively involving in the making a force on the foreign or special or our own cells. The making force on the cells is the major objective of this major aim of this complement system. By by using different branches of the immunity, whether they can humoral immunity may be involved in this type of complement system, or cell mediated immunity may be involved in this complement system. By using these two branches, this complement system makes a force on the foreign or our self cells, infected or infectious cells. Okay, viral infected cells or tumor cells, which are not used, which are harmful for our body. The system recognizes those type of cells, and with the help of complement system, those harmful cells can be eliminated by using complement pathways. Okay, these are aspects we have discussed in the earlier classes. Okay, up to second block. Up to second block, we have discussed in our offline classes, and these online classes I am going to tell you about. I am going to start from transplantation, that is block 4 unit 14. Okay, transplantation and organ rejection. Okay, uh, you know about the transplantation. uh, just you have listened this type of terminology in the newspapers and TV news. Okay, and transplantation or grafting, we can also call as grafting, and is the transfer of cells or tissues or organs. Okay, what is that? Transplantation is the transfer of cells or tissues or organs from one person or one part of the body or one person to another person, one part of the body to another part of the body. That is the transplantation. And in this, two members or two persons are actively involved. One person can be called as donor. Okay, another person can be called as uh, recipient. Okay, I will show you one image for that. And different types of organs, whenever the uh, damages or dysfunction of the organ can be seen in the person, those organs are replaced with the help of transplantation technology. Heart can be replaced whenever these heart vessels or heart valves get damaged. And eye cornea and lung transplants can be possible, and tracheal transplant can be possible, liver transplantation, and kidney transplantation frequently. You most of the times you have heard about this kidney transplantation and heart transplantation especially. And pancreas transplantation can be possible, and skin transplantation whenever the bones and accidents in the accidents, the skin rupture, this type of transplantation can be seen. And vascular tissues transplantation can be seen in the terms whenever the blood vessels are get damaged. Blood vessels can be transplanted or replaced with the tech by using transplantation technology. So simply, um, the donor from the donor, the recipient will receive an organ, and this the those organ can be called as graft. Okay, organ, especially organ, can be called as, or transplant can be called as graft. Okay, which is a one specialized organ. Okay, and fate of the graft is determined by the genetic relationships. Very very important point is that genetic relationship should be seen, can be matched with the donor and recipient. What is genetic relationship? The genetic relationship in terms of the matching of uh, tissue or cells, surface receptors. Especially, we have talked about major histocompatibility complexes. What are they? The major histocompatibility complexes are cell receptors. Those are two types, especially. One is that one is there on the all types of cells. I will show one uh, uh, slide for you. You can then only you can recognize what is major histocompatibility complexes. Uh, 1 minute. Yes. No. Major histocompatibility complexes. Anyone from the group? Do you know? Do you know the major histocompatibility complexes? What are they? Anyone from the group?
Can you repeat
What
the
is
question?
major? Tell me.
Please repeat the question.
MHC1 is
What?
present on cell surface and MHC2 will be active, that is in intracellular.
Mm.
mediated immunity.
Okay, okay, very nice, very nice. For your reference, I have shown on the screen. Just see the screen. The major histocompatibility complexes or receptors present on the cell surfaces. Okay, whenever our cell get infected by the parasites or pathogens, our cell show the major histocompatibility complexes with the effect of uh, with uh, uh, with against the parasites or pathogens. Whenever our cell get infected with the parasite or pathogens, and information can be taken up by this receptor and presented on the cell. If the MHC molecule is present on the cell surface, signal the meaning of this presence of the MSC molecule is that the cell is infected by the different pathogen or parasite. Okay, these MSC molecules are two types. Okay, if all the times MSC molecules are not present on the cell surface, whenever our cell get infected by the pathogen or parasite, whether they may be endogenous parasite or antigen or exogenous antigen, our cell MSC molecules collect collect those antigens. Are you following me? Our MSC molecules are there in our cell, not on the receptor. Whenever we not encounter with the any pathogen or parasite in the normal case, whenever we get infected, our cell get infected by the pathogen or parasite. Those uh, pathogen or parasite are digested with the help of our lysosomes. Then the active antigen molecules are collected by these MSC molecules. MSC molecule can collect these processed antigens from the cell and present on the cell surface. The antigens can be presented with the help of MSC molecules on the cell surface. Not all the times, but infected times. Only MSC molecules are exposed or present on only especially infection times. Okay, and MHC1 molecule is there all the found all the surface of the nucleated cell. Point 4. So you can see this term in the point in the point 4. Okay, as point 4 found on all surface of nucleated cell. The very important point for your transplantation immunology. And coming to the MHC molecule 2, found on surface of antigen presenting cells and activated T cells. See, the main difference between MSC molecule 1 and 2 is that MSC molecule class 1 present on all the surface body cells. Okay, but MSC molecule 2 is only present on antigen presenting cells. Those are professionally acts as a antigen presenting cells. The function major role of the cells those cells are modified for the antigen presenting property. For example, our T cell cannot recognize the antigen directly. Okay, B cell has a capability to detect the antigens directly, but our T cell, major acquired branch of the immune branch cell, is the T cell. The T cell is a major cell, but it has a no, it don't recognize the antigen directly. But with the help of antigen presenting cells, it recognizes the antigen. Okay, uh, antigen presenting cells, for example, dendritic cells, macrophages cells, different types of antigen presenting cells are there. Those except our body cells. Okay, these are the uh, wanderings, patrolling cells. Antigen presenting cells are patrolling cells. But coming to the MHC molecule 1, our all surface cells, nucleated cells are having MHC molecule 1. Okay, remember this point. And graft, coming to the graft, okay, coming to the graft, the Different types of the grafts are there. Just remember this point. This is very very important point before going to know about the graft rejection. Okay. Coming to the graft, what is graft? Transplant. Okay, transplant. Then synonym of the transplant is that graft. Okay. Uh, Transplantation types, graft types, transplant or graft types of grafts. Uh, I'm going to show you one image for image. Just a minute. Yes, the types of transplantation. Okay, the types of transplantation. Coming to the types of transplantation, graft. That means graft from one person to based upon the transplantation from one person donor to recipient. Okay, uh, autograft. One type is that autograft. Autograft is self tissue transferred from one body site to another. Is in the same individual. Um, uh, in the same individual, okay, self tissue transferred from one body site to another in the same individual is called as uh, autograft. Okay, autograft means self transplantation. Okay, graft transplantation. Transferring healthy skin, one example, I am giving one example. Transferring healthy skin to a burned area. Whenever we get a fire accident, our skin may be damaged or get burned. Then at that time, our damaged burned area can be replaced with healthy skin by using this transplantation technology. This type of transplantation is called as autograft. Auto means self. Okay, as shown in the figure. Coming to the isograft. Okay, I think isograft is not there. Uh, Syngenic. Okay, syngenic graft means in the twins. Okay, in the twins. Syngenic means in the twins. And allogenic. Allogenic is tissue transplantation. Syngenic means I will explain some more. Syngenic means that can be defined as a isograft. Okay, uh, transferred between genetically identical individuals, twins. For example, twins. Transplantation takes place between twins, identical twins. That type of transplantation is called as isograft or syngenic transplantation. Coming to the allogenic, okay, allogenical allograft is the tissue transferred between genetically different members. Okay, genetically different members of the same species. The same species, the donor and recipient should be same. Should belongs to the same species. For example, Homo sapien. Okay, should be same species. Uh, Transplantation between uh, same species can be called as allogenic. Organ transplantation between the same species. Okay, genetically different members, not identical. If there is identical, that is syngenic. But if there is not identical, allogenic. Okay. Coming to the xenograft. The xenograft is one another type of the grafting. What is xenograft? Xenograft is tissue transferred between different species. Okay, as shown in the figure, the pig graft transplantation between pig and man can be called as xenograft. Okay, it can be possible. A xenograft is tissue transferred between different species. Okay, because of the significant shortage. Okay, what is the reason behind it? Significant shortage in donated organ. Whenever the organ donors are not there, or we can collect the organs from the cultured pigs or monkeys and baboon, etc., etc. And this type of the graft is called as xenograft. These are the types of grafts. Okay, types of the graft. What is graft or what is transplantation, and types of transplantation, types of grafts. Okay, laws, transplantation laws. You can see in your book, page number 201, 201. all the information has given in the 201 page. You can see your page number 201. Whenever the major transplantation laws are there, okay, transplantation technology uh, depends upon the few laws. What are they? The five laws, major five laws are there. Transplantation within inbred strains are accepted. Okay, that is syn graft. We can call it isograft. It can be accepted. And transplantations between inbred strains, okay, are rejected. Allograft, okay, are rejected. Between inbred strains are rejected. And transplantation from either inbred plant parental strains to F1 hybrid are accepted, but transplantation in the reverse direction are rejected. F1 can be acts as a donor, but reverse cannot be happen. Transplants from F2 or later generations animals to F1 animals are accepted. F2 later can be accepted. Transplant from either parent strains are accepted in some F2 animals but fail. All these laws are based upon the cell surface molecules. Cell surface molecules can be matched, or genetical makeup can be matched with the donor and recipient cell surface molecules. Okay, especially class 1 MHC molecules and class 2 MHC molecules. Okay, before going to talk about MHC molecule play a role of the MHC molecules in the graft or transplantation technology, I would like to talk about rejection. What is graft rejection? We have talked about what is graft, or what are the types of the grafts are there, how many types of grafts there are there. And now I would like to talk about rejection, transplantation rejection. Okay, what is exactly the transplantation rejection? I will show one image for you. Rejection, the main term itself indicating that rejection, okay, rejection. Transplantation sometimes can be rejected, or graft can be rejected by the immune response against the graft. I will I one point I will say, uh, the mechanism is the same. I will show one image for you exactly what exactly our body can do. Our body always prevent the entry of foreign particles. Okay, our body can always prevent, or can fight always against the foreign invaders like pathogen and parasites. In this type of uh, defense system, our self immune system recognize as a foreign particle. Graft can be recognized as a foreign material. Whenever our body having a graft on our body, or in the body as a organ, our immune system recognize it as a foreign particle or foreign material or foreign pathogen or parasite. The fate of graft is determined by the genetic relationship between donor and recipient. Grafts are generally accepted, generally some accepted if the genetic makeup and histocompatibility antigens are same.
Excuse me, sir, your voice is not audible. It's not clear, sir.
Hello, am I audible now?
Yes, sir.
Yes, that is a signal problem, network problem. So, uh, whenever the graft generally accepted when genetic makeup and histocompatibility antigens are same on both recipient and donor should be matched with a recipient surface antigens or cell surface antigens should be matched with the donor surface molecules or antigens, and then only the graft can be accepted. When this graft can be rejected, when there is immune response against histocompatibility antigens. Whenever the donor surface antigens are different from our recipient surface antigens, our recipient immune system recognize as a foreign molecule or foreign pathogen or foreign parasite. In general, whenever our body encountered by the different pathogen or parasite, our immune system recognizes by the studying its surface molecules. With the help of our innate and adaptive immune system, they can be the antigens or foreign pathogens can be eliminated. Same like that, whenever our body uh, uh, transplantation has having transplantation or graft, this graft should be matched with our cells or cell surface molecules. Then only our body accept this graft. Our immune system accept this type of graft. When the surface histocompatibility antigens are not same, then our body rejects, our immune system fight against those graft. Then this type of rejection, this is called as rejection. Depending upon the speed of the rejection, graft rejection are four types or of four types. Okay, they are four types depending upon the speed of rejection. One is that hyperacute rejection, name itself indicating that rejection of graft within 1 hour or minutes, within few minutes, within 1 hour, that type of rejection is called as hyperacute rejection. This is one type of rejection. And coming to the second type of rejection, accelerated rejection, when our when the graft rejection takes place from the first day to 4 days after grafting. This type of the rejection is called as accelerated rejection. Okay, and third one is that acute rejection. This type of rejection can be seen within a month, after a month, or within a month, just within the month period of month, organ can be or graft can be rejected. This type of the rejection is called as acute. Acute means faster, and chronic means slower. Okay, chronic, you have heard about chronic diseases like tuberculosis. Okay, chronic means long time period, long period. Can be seen slow and gradual rejection within a week or months. It will take months of time. Okay, 1 year, 6 months, 1 year, up to 6 months, up to 1 year. Organ can be rejected. Okay, when the organ can be rejected, the tissue surface molecules are not same. Okay, those type of molecules can be those type of tissue can be rejected from our body. Okay, our immune system will be get activated by the foreign cells surface molecules. Whenever those surface molecules are not same, then it fight against foreign grafts. Okay, that is the phenomena. And coming to the MHC molecules, okay, if you understand about the MHC molecules, you can understand entire things about transplantation. The role of MHC molecules in the rejection. Okay, immunity. You can call as immunity. Role of MHC molecules. Okay, I will show. Yes. The role. What is the role of MHC molecule? MHC molecule one, major histocompatibility complexes are expressed on the surface of nearly all nucleated cells, MSC molecule 1. The major function of the class 1 gene products is presentation of peptide antigens to CD8 T cell. Okay, T cells are okay. T cells are of three types, majorly two types. Okay, don't bother about third type. T cell, T H cell or helper cell, T cytotoxic cell. Another one is the T suppressor cells. Don't bother about this third type. Just you uh, know about the know more about TH cell and TC cell. Okay, TH means helper cell. C means cytotoxic cell. Okay, um, major histocompatibility cells or one complex one present on all the cells. The function of this cell is that presentation of peptides, and those are the antigens, okay, foreign antigens to CD8 cell. Okay, what is CD8 T cell? TC cell contains CD8 receptor, a clustral differentiation receptor on the surface of T cell. Okay, this TC cell contains CD8 type of markers on the surface of the cell. Okay, all the times MSC molecule 1 binds with the TC cell only because it has a matching system. Okay, with the TC cell with the CD8 marker. Okay, TC cell. And function of T coming to the MSC molecule 2 is also one there on the surface of the antigen presenting cells. Okay, our every cell acts as an antigen presenting cell when our body encountered by the pathogen or parasite. All the cells are having MSC molecule 1. Especially our in the blood circulatory system or lymph circulatory system, the specialized cells are modified for the antigen presentation. Okay, example, macrophages, dendritic cells, B cells are modified for the antigen presenting property. Those are called as professional antigen presenting cells. Okay, and these cells collect the antigens, okay, process the antigens and present the antigens with the help of MHC molecule 2. Okay, this MHC molecule 2 is a receptor, a surface molecule, and binds always with binds always with CD4 marker on the TH cell. Okay, CD4 is a clustral differentiation marker present on the TH cell, T helper cell. MSC molecule 2 binds always with the TH cell. MSC molecule 1 binds always with TC cell. This is a principle. Okay, and coming to the coming to the graph rejection. Okay, it plays a major role. MHC molecule 1 play a major role in the antigen presenting cell. Whenever our body cell get infected by the viral cells, okay, viral cells or bacterial cells, endogenous pathogens or antigens, this MHC molecule collect the uh, those our cells process the uh, foreign particles, and those process the antigens present presented by the MSC molecule 1. What happens after the presence? After the presence of the MSC molecule with the antigen on the surface of the cell, infected cell, the fighting against the foreign particles will be started. When the antigen after the antigen presentation, okay, and what happens? The T cell come and contact with the MHC molecule 1 with the antigen. It binds. Okay, I will show one example, one image for you. Okay, that is cell mediated immunity. That is comes under cell mediated immunity. For the reference, you can study about cell mediated immunity. I will show what is exactly happening. 1 minute. Okay. Yes. MSC molecule. Whenever our uh, body get encountered by the foreign particles, see our antigen presenting cell present the antigens, and these antigens present with the help of MHC molecules. Whenever the MHC molecule present on the surface, our that is the signal for the T cell attraction. Okay, chemo attraction can be seen. This TC cell goes and binds with the MHC molecule with the help of CD8 marker. Okay, CD8 marker, and this the communication between the CD8 and MHC molecule can be trigger the activation of the TC cell. Okay, this TC cell get activated. Okay, after the activation, the molecules can be released. Okay, that is the great uh, phenomena, and this molecules, specialized molecules, can be released from the activated cells, and these activate molecules against activate the further activate the TC cell. After the full activation, TC cell uh, undergoes the proliferation. TC cell undergoes the proliferation and differentiate into the two cells. One is the memory cell and another one is effector cell. Effector cell, whenever the effector cell is generated, the effector cell will release the most dangerous cytokinins, enzymes, pro granzyme. Okay, like those enzymes, destructive enzymes. Those enzymes are released on the surface of the infected cell or the uh, pathogen and parasite. Okay, those enzymes or molecules are most dangerous or effectively degenerates the plasma membrane of uh, either the pathogen or our infected cell. By those pathway, our MSC molecule play a key role in the graft rejection. This is the general pathway for any infection. Well, our graft rejection process is same like our infection. Whenever our body get infected by the pathogen or parasite, same pathway can be seen in the time of rejection also, graft rejection also. Okay. Coming to the MSC molecule 2, hey, this assume that this is our antigen presenting cell, okay, which is having MHC molecule 2. Okay, specialized cells are on the surface of specialized cells. MHC molecule 2 is present. Every body cell, MHC molecule 2, MHC molecule 1 present on all the body cells, but not MHC molecule 2. And whenever our body get encountered by the pathogen parasite, these macrophages or dendritic cells, B cell, B lymphocytes recognize the antigens and process the antigen by engulfing them. And after the process, these MSC molecules, uh, antigens are presented by the MSC molecule 2. This is MSC molecule 2. And whenever the MHC molecule 2 present on the cells are present on the antigen presenting cells, those are professional antigen presenting cells, TH cell attracted towards the MSC molecule 2 having cells, okay, 2 present cells, okay. And this TH cell uh, goes towards the MSC2 molecules and binds with the MSC2 molecules, which is having the antigens, and the contact between these two, MSC molecule 2 and CD4 marker, okay, CD4 marker, it can be trigger the activation of the TH cell, okay, TH cell, and this activated TH cell undergoes the proliferation. One is the memory cell and one is the effector cell. The effector cell uh, can bind with the B cell, okay, or either T cell, and these two types of property can trigger the furthermore activation of the TH cell and B cells. Okay, whenever the TH cell activation is there. Whenever the Th cell get activated, undergoes the proliferation. This is all is about the cell mediated immunity, not a new one. Okay, we have discussed in the cell mediated immunity. As part of our cell mediated immunity, entire things are discussed in the cell mediated immunity. Okay, just I am revising that. Okay, activated Th cell get proliferated, and one effector cells goes and binds with TC cell or B cell, and binding between the Th cell and TC cell or B cell furthermore activated the TC cell or B cell. And this, if the Th cell activate the TC cell, this TC cell undergoes the proliferation. And same matter, one cell is memory cell, another one is effector cell. And effector cell will release the enzymes, granzyme. Okay, uh, different types of lytic enzymes. That is the phenomena. And if the B cell is activated, this cell also uh, get activated. Whenever this B cell get activated, undergoes the proliferation. When one cell is the memory cell, another one is the effector cell or plasma cell. Okay, the plasma cell is a specialized cell which is having the property to release the antibodies. Okay, these are the antibodies. Okay, immunoglobulins, we can also call as immunoglobulins. And release the immunoglobulins goes and binds with the uh, antigens. Okay, antigens and neutralizes the antigens. This is the uh, phenomena what is happening in the graft rejection. Okay, what is the main role of the major histocompatibility complexes? After the releasing of the uh, antibodies, another different types of immune pathways are get activated. For example, complement system get activated after the releasing of the immunoglobulins from the B cell. Okay. Next, another pathway is that uh, antibody dependent cell mediated cytotoxicity is one of the hypersensitivity reaction that is also get activated after the releasing of the antibodies from the B cell. The many types of the pathways are get activated after the contact between the Th cell between and major histocompatibility cells, that is antigen presenting cell. This is how the major histocompatibility complexes are play a key role in the graft rejection process. Okay, the the role of the graft rejection, role of the major histocompatibility complexes uh, in the graft rejection is the very important. Okay, uh, uh, are you following me? Any doubts? Any doubts?
Okay.
No doubt, sir.
Okay. This is the one of the pathway what we have talked in the cell mediated immunity and humoral immunity. If you learn about the cell mediated immunity and humoral immunity, you can read everything in the immunology in your book. Okay, that is the very important. What is that? Humoral immunity. Another one is that cell mediated immunity. Just understand those things very excellently after the reading of the humoral immunity and cell mediated immunity. After understanding the after understanding of these two things, go for further topics. Just you cannot read another topics without these two topics. We cannot separate these all the topics from the cell mediated and humoral immunity. Okay, and another one is that the rejection. Okay, rejection mechanism we will discuss tomorrow. Okay, in the next class. What is rejection? What is rejection mechanism? What are the types? What are the cells involved in the rejection mechanism? We will talk later. Okay, if you have any doubts, just ask me. Okay, this is the introduction class. We have discussed about transplantation. Okay, what is transplantation? Okay, what is graft? Okay, and types of graft we have discussed. Okay, that is there in your 201 page. Autograft, what is autograft? What is isograft? What is uh, allograft? What is xenograft? Okay, we have discussed about loss, and we have discussed about MSC molecules. Okay, and we have discussed about rejection types. Okay, depending upon the speed of rejection, what is rejection we have discussed, and we have discussed about rejection types. You have two types of, majorly two types, that is acute rejection and chronic rejection. But in your book, they have mentioned four types of rejection: hyperacute rejection, accelerated rejection, and acute rejection, chronic rejection. Okay, majorly there are two types of rejections are there: acute and chronic rejections. Okay, we have discussed everything, and after that we have discussed the role of MSC molecules. Okay, the cell uh, histocompatibility complexes are there in every uh, organism. And coming to these molecules, we have two types of MSC molecules are there majorly. Class 1 MSC molecule is there on every nucleated cell, but class 2 is there on only specialized cells. Those are called as professional antigen presenting cells. For example, macrophage, dendritic cell, and B cells. These are three cells are called as professional because they are the profession of these cells is that presenting the antigens to T cell. Okay, T cell, especially T cell, and this after the processing of the antigen, our body cells. Okay, uh, MSC molecule, by with the help of MSC molecules, antigens can be present to the T cells. Okay, whether TC cell, whether it is T cell or TC cell or TH cell. If it is TC cell, the damaged or infected cell directly, TC cell directly damage the infected cell. But if it is TH cell, it cannot damage it directly the infected cells or tissues. With the help of whenever the TH cell get activated, it cannot damage the infected tissue directly, but it activates the TC cell and B cell. And after the activation of the TC cell, then only the infected cell get damaged. If the B cell is activated with the help of TH cell, this activated B cell releases the antibodies. Okay, these antibodies, from the antibodies, many types of pathways are activated. I will show one example for you. Different types of pathways are get activated. This is how our uh, different types of pathways are involved in the rejection, graft rejection. Okay, for example, lytic enzymes. Okay, this is graft. Again, these are the mechanisms. Okay, fight against the graft. Okay, graft nothing but a, if our body immune system accept the graft, no issues. If our immune system reject the graft, these by all the mechanisms, our immune system get encountered about the graft. Okay, for example, whenever our B cell get activated, this cell uh, undergoes the proliferation. One is memory cell, another one is this plasma cell. Okay, this is plasma cell, and plasma cell will release the antibodies. These are the antibodies, goes and binds with the graft tissue surface molecules, and this graft, whenever the antibodies goes and binds with the tissue surface molecules, that is antigens, and these complexes, antigen antibody complex, will be found, and these complexes will trigger the further mechanisms. Okay, for further anti-immune mechanisms. For example, ADCC means antibody dependent cytotoxic, cell mediated cytotoxicity. Antibody dependent cell mediated cytotoxicity. The complex between formation of the antigen antibody complex will trigger the different types of immune reactions. Okay, pathways get in different types of immune pathways get activated after the formation of immune complex between the antigen and antibody. Okay, uh, a complement system get activated after the formation of the antigen antibody complex. Okay, and membrane damage will be takes place of the one of the pathway, and MSC express will trigger the different types of pathway, and uh, these CD8 cell, CD4 cell, B cells, all these cells, T cell and B cell actively involving in the graft rejection. Okay, we will discuss pathways. Okay, steps. Okay, in two steps, first set of rejection and second set of rejection. The two in the two steps, uh, graft can be rejected. What are they? We will discuss in the next class. Okay, we will discuss later in the next class. If you have any doubt,
Okay, sir. Thank you.
you can ask. Okay, just read out the page numbers from the 198 to 201. Okay, we will discuss of later pages tomorrow. From the
Okay, sir.
2022, after 2022, we will discuss. Okay, all the rest. Bye.
Okay, sir. Thank you.
Ah.